Proteins and enzyme inhibitors, AQA biology. As we begin more videos, I'm going to start with key terms. So you have proteins, which are large molecules that are very similar to each other. The only really real way that proteins differ from each other is with the R groups, but we'll get onto that. A peptide bond is formed by a condensation reaction which joins amino acids together, and amino acids form proteins. A dipeptide bond is two peptide bonds. Chromatography is a separation technique that relies on components' affinities to the mobile and stationary phase. Now the mobile phase would be like the solvent that runs up the stationary phase, and the stationary phase in terms of, let's say, paper chromatography would be the paper. It's the thing that doesn't move. Globular proteins are proteins with a function, and fibrous proteins are molecules that have a structural component, for instance, cellulose. Enzymes are globular proteins that act as catalysts. And a biological catalyst is a soluble protein molecule that can increase the rate of reaction without being used up. Now, the activation energy is the minimum amount of energy that the reactive species must have in order to undergo the reaction. The induced fit theory is that the active site isn't 100% a match for the substrate. Like, it's similar enough that when the substrate binds, the active site changes shape slightly and allows the enzyme substrate complex to form. Cofactors for enzymes are some enzymes need a non-protein substance before they can actually catalyze a reaction. So that's what that is. And then coenzymes are organic molecules which bind temporarily to fit the enzyme, transferring a chemical group necessary for the reaction to occur. Enzyme inhibitors prevent the enzyme activities. Competitive inhibitors are similar shaped to the substrate, and non-competitive inhibitors, that is a hard word to say, bind to the enzyme away from the active site. This binding causes the active site to change shape so the substrate can no longer fit. Proteins. Now this is the general structure of a protein molecule. So all protein molecules will have the amino group, which is highlighted in red, the carboxylic group, which is on, like, you have the line underneath, and then you have the side chain as well as the hydrogen. So the amino group, the hydrogen, and the carboxylic acid group are always there. The only differ is the R group, like I mentioned before. But when you're drawing your amino acid molecules, you can literally just write R unless they specify differently in the question. Now, the test for proteins. Proteins are tested using burette solution, and a positive test will turn the solution from blue to purple. The different types of chromatography are paper, thin layer, and gas. Paper would be, you get the paper and you draw a line at the bottom of the paper, not completely at the bottom, maybe 1 or 2 centimeters above the bottom. Then you, for instance, if you wanted to find out the solubility of an ink, you put a dot on the middle of the paper. Then placing it in a solution below the line in which you drawn, allow the solution to run up the paper and make sure you take it out before it hits the top of the paper. Then you draw a line to where the solution had stopped running, and that will be your solvent front. Thin layer chromatography is a bit like that, but it requires few more steps, but that's more chemistry than it is biology. I haven't really seen a question come up on it. And then gas chromatography, as well as thin layer, is more so chemistry. I haven't really seen a question on it. However, the thing that you're measuring in gas chromatography would be the abundance collected instead of how far the dot or the substance has traveled. RF value is calculated by the distance moved by the spot divided by the distance moved from the solvent front. Now these are your 4 different types of protein structures. You have your primary chain, which is basically your polypeptide chain, and if you don't really know what a polypeptide chain is, I suggest you looking at my biological molecules video. The secondary structure, which is either an alpha helix or a beta pleated sheet. Now these structures are held together by hydrogen bonds. A tertiary structure is also held together by hydrogen bonds, but it allows the molecule to fold in on itself, forming ionic bonds, disulfide bridges, and such. And then a quaternary structure is when you have four tertiary structures joining together to form one molecule, and sometimes they'll have a prosthetic group. Now, what are the different types of protein DNA codes for? Well, you have enzymes, antibodies, transporter proteins, and structural proteins. Enzyme action. Now, I've only done pictures for these because I feel like, at least for myself, I learn better from pictures. So what you have in the beginning, you have your enzyme and your substrate. Now, as you can see, the bottom of the substrate is a similar shape to what the enzyme is. So it's able to bind to that enzyme, forming an enzyme substrate complex. So now that enzyme substrate complex is then broken down by the enzyme to form two products in which break apart once they are formed. However, and that is called the lock-in key. Now the second type is called the induced fit model. And I mentioned this at the beginning of the video. Now, the substrate entering the active site, as you can see, they're not 100% matched like the first picture shows. However, the enzyme is able to adjust slightly and change shape as the substrate binds, forming your enzyme substrate complex. The enzyme produces the products, and then the products leave the site of the enzyme. Factors affecting enzyme action. How are enzyme catalyzed reactions measured? Well, you can either measure it by the formation of the products or the disappearance of the reactants. And how rate of change is measured is using a calibration curve, like is shown in the right-hand side corner. You pick a chosen point and then you work out the gradient to find the rate of reaction. If it's a curve like this, then you might want to draw a tangent line. The effects of temperature. Enzymes vibrate more at a higher temperature, which causes more collisions to occur, forming more enzyme substrate complexes. However, above 40 degrees, enzymes denature as ionic bonds in the active site break. Now, this breakage causes the active site to change shape so that the substrate can no longer form and no longer bind, which means no enzyme substrate reactions can take place. Now, the effect of temperature. Now, the optimum pH for most enzyme reactions is 7. However, if it's like an enzyme in the stomach, obviously the pH of the stomach is pH 2, so obviously it work better in a pH of 2. However, that's the most accumulative pH that's in your body overall. Ionic bonds are altered by changes in pH, and this breakage of ionic bonds changes the shape and denatures enzymes. Effect of substrate on enzyme concentration. Effect of substrate concentration on enzyme action. So sorry for that. Greater collisions between the substrate and enzymes, and this happens until all of the enzymes are full. But if all of the enzymes are full and blocked, that means no further reaction can take place until they are freed up. So substrate no longer has an effect, as enzymes are now a limiting factor. Enzyme inhibition. So now you have two types. You have competitive and non-competitive. And competitive inhibition, you'd have a substrate and an inhibitor. Now the inhibitor and the substrate have a similar shape. So if the inhibitor binds to the enzyme instead of the substrate, it efficiently blocks the active site, which means no reaction can take place. Therefore, you have all this substrate in space, but all of the enzymes are blocked, so no reaction can occur. And then you have non-competitive, in which the normal substrate can still bind to the active site until a non-competitive enzyme inhibition comes. And when it binds to the side of the enzyme. And this binding causes the active site to change shape. So as you can see here, whereas before it was a perfect fit for the substrate, Now the substrate no longer fits it properly. And as it doesn't fit properly, it means the reaction can no longer occur. Now, hopefully all of that made sense. And the next video, I'll actually be going over nucleic acids, so such as the structure of RNA and DNA, as well as DNA replication and evidence for DNA replication. Thank you for listening. If you like the video, then please subscribe and comment, and see you next time.